So I'm Arturo Molina. I'm Chief Medical Officer at Protagonist Therapeutics. I'm also a volunteer adjunct clinical professor in the Department of Medicine, Division of Oncology at Stanford University. And I'll be talking about the potential role of the hepcidin mimetic Rusfertide in polycythemia vera. So it's already been discussed that PV, polycythemia vera, is characterized by abnormal RBC production, iron deficiency, and thrombosis. And one of the reasons I highlight iron deficiency is that sometimes that is underappreciated, the impact of iron deficiency, that is. And yesterday and today, there's been a discussion of the symptoms that can be associated with PV, which include fatigue, dizziness, pruritus, problems with concentration, also known as brain fog. And if you look at the part of the slide that says abnormal laboratory abnormalities, I also draw your attention that red blood cells that are being produced in polycythemia vera are abnormal, in part because they are being produced in the setting of iron deficiency. And the iron deficiency occurs because all the iron's being used up for the excessive red blood cell production. But why does the iron deficiency get worse in PV? Phlebotomies. Every time you get phlebotomies, you're losing iron. So not only, okay, so not.
You have to eat it.
You have to put it right in my face.
Yeah.
Okay, no, but not only is the bone marrow using up all the iron and producing too many red blood cells, but also the treatment for PV involves removing iron because you're removing the red blood cells. So you get like a double hit in the iron deficiency. So you've already heard a lot about the treatments for PV. I'm not going to go through all of them, but phlebotomy is one of the main treatments for PV. And one of the things about phlebotomy, it works pretty well, but for many patients, they go through this roller coaster where the hematocrit goes up to 48, 50, then you get a phlebotomy, it comes to 45. For some patients, that'll work for a month, for some 2 months, for some a few weeks. And as was mentioned, some patients know when their hematocrit's going up again. And so the hematocrit goes up again, and that triggers a phlebotomy. But what is not happening is the hematocrit staying below 45 continuously, consistently, 24/7. So that's where I think Rusfertide might have a role in the future. So as I mentioned, frequent phlebotomy induces or worsens iron deficiency and exacerbates the symptoms, including dizziness, headache, and weakness. And then in some reports, when patients are surveyed, 55% of patients report that phlebotomies pose a burden to their overall quality of life. But doctors also think that phlebotomies are burdensome for patients. And this data is mainly from the US, but the median time spent in a phlebotomy is about 4.2 hours. I think in some places in Europe, I think it's a longer procedure, but it does inconvenience patients, and it has limitations because it only works while the hematocrit's down, but the hematocrit always comes up if phlebotomy is the main way that the hematocrit's being managed. So I'm not going to go through this list, but I do want to make a plug for phase 3 studies, and you'll have the slides to look at. But this is a listing from clinicaltrials.gov of phase 3 studies that are ongoing. You'll notice that many of these studies involve compounds that are already approved, including one study that's comparing ruxolitinib in the frontline setting compared to hydroxyurea and interferon. But the only new compounds that are being tested that are unapproved in phase 3 studies are Rusfertide, that I'm going to talk about, but also a compound called givinostat. And I think this study is mainly being conducted in Italy. But again, This is also a good time for me to thank all of the patients who participate in clinical studies, because without your participation, we wouldn't be able to get the knowledge that is needed to get the drugs approved and bring them to a broader patient population. So what is the rationale for using a hepcidin mimetic? I'm going to come over here and point a little bit. This is the iron that's circulating, that's being fed to the bone marrow. And in polycythemia vera, there's a low hepcidin state. So what does hepcidin do? Hepcidin binds to ferroportin. Ferroportin is the transporter of iron outside of macrophages. So when the hepcidin is low, iron is being delivered to the body and stimulating the erythrocytosis in the bone marrow. What rusfertide does and what hepcidin does, it binds to ferroportin, decreases the iron, decreases erythrocytosis. And so one way of thinking of rusfertide, it's like a hormone, like a thyroid hormone. Well, thyroid thyroid hormone replacement controls the thyroid function. Well, hepcidin is the hormonal regulator of iron metabolism or iron homeostasis in the body. And this becomes an important concept because here I'm showing you that hepcidin is blocking iron, but I'm also going to show you some data that hepcidin over time increases ferritin, which is a protein that carries iron. And so maybe what the compound is doing is preventing iron delivery where it is not needed, but maybe allowing some iron to still go to other parts of the body where it is needed. So rusfertide is being investigated in phase 2 and phase 3 studies. I'll start from the bottom. The Pacific study was a study done in South Korea and Indonesia, and it was for patients who had uncontrolled hematocrit. So all these patients had to have a hematocrit of 48 or higher. And what we showed there is that within 4 weeks of rusfertide treatment, the hematocrit comes down to the normal range. I need to mention that rusfertide is an injection that's given once a week. The patient initially the injection's given usually in the office, but patients learn how to self-administer, and the majority of injections are given by the patient themselves. Then there's the REVIVE phase 2 study. I'm going to focus quite a bit on that study. And then VERIFY is a phase 3. We just completed enrollment. The target enrollment is 250 patients. And on the left side of the slide, you'll see that under Protagonist Therapeutics, Takeda is mentioned. And the reason Takeda is there is that we recently partnered up with Takeda. Takeda is a much bigger company. Protagonist, we're in California. We're about 120 employees or so. We're still responsible for all the clinical development of rusfertide, including regulatory submission. Takeda will be responsible for commercialization. And they may be involved in filing in Europe. That still remains to be determined. Or regulatory filings outside of the United States. And later I'll tell you a little bit about THRIVE. I'm not going to get into it right now, but I'll keep going. So REVIVE is a phase 2 proof of concept study of rusfertide for excessive polycythemia vera associated erythrocytosis and hematocrit control. So there are 3 parts to the REVIVE study. And I should mention the REVIVE study was done mainly in the United States and a few sites in India. So in part 1 is the dose finding. Patients start out at 20 milligrams. They titrate upwards until the hematocrit is under control. Then there's an efficacy evaluation from week 17 to 28. And then part 2 is a blinded withdrawal. So this is different than VERIFY. In REVIVE, everybody started out with Rusfertide. And then when they go to part 2, half the patients stayed on Rusfertide, the other half went on placebo that looks just like the Rusfertide. And then part 3 is an open label extension part. And one thing that's different from this study is that all patients need to get a phlebotomy or phlebotomies to bring the hematocrit below 45. So for Revive and Verify, the hematocrit is 45 when patients start the treatment. The Pacific study that I mentioned before from South Korea and Indonesia, all these patients had uncontrolled hematocrits. They were not phlebotomized to below 48. And the main rationale is that the FDA wants all the patients to start with a hematocrit that's well controlled to test the effect of the drug. So this is a very busy slide, but I'm just going to make a few points from here. There's a predominance of males. The median age at the time of a study entry was 58, and about half the patients are high risk, half the patients are low risk. And if you look at age at diagnosis, the median age is 55, but one of the patients had PV diagnosed at age 5. And this kind of reminded me of the question that was discussed yesterday, that there's very little data on kids who get myeloproliferative neoplasms, but some of these patients who have had an MPN when they were young do sometimes get to participate in clinical trials when they get older. And then important to note that for the Revive study, in the 28 weeks prior to starting treatment, the median number of phlebotomies that they required was 4.7. One other point, because that's a very important point. If you look at the concurrent therapy, 52.9% were on phlebotomy only, and the other half of the patients, 47%, were on phlebotomy and cytoreductive therapy. And the cytoreductive therapies were mainly Hydrea, but some patients were on interferon, some patients were on ruxolitinib, and a few patients were on interferon and ruxolitinib. This is an add-on treatment, so it can be used with phlebotomy only, or it can be used as a combination with Hydrea, interferon, or ruxolitinib. And from the phase 3 trial, we're going to have a lot more data on the combinations. So this is the part 1 of the study. And what you see in the figure on the left is for patients who were only on phlebotomy. The figure on the right is for patients who are on phlebotomy and cytoreductive therapy. And then right here is when the treatment started, the dose 1. So every little red triangle represents all the phlebotomies that the patients had received in the previous 28 weeks. And then what you see after starting treatment, what happens to all the red triangles? Well, you don't see them anymore in most of the patients. So most of the patients do not require phlebotomies after starting treatment on Rusfertide. So this is in the part 1 of the study where everybody got Rusfertide. Now, the Rusfertide was on a clinical hold. This is the gray part here. There was a finding in a preclinical model of carcinogenesis where in these mice, there were some skin lesions. Most of them were benign papillomas, but in each group of the animals, there was a squamous cell carcinoma of the skin. And we had to amend the informed consent forms to just make the patients aware of a potential risk of cancer. And what you see when the drug was stopped in the gray area is that the need for phlebotomy returned right away. So even before we unblinded, during this temporary suspension of treatment, we got a strong signal of the efficacy or the lack of efficacy when the drug is stopped. So this is the result from part 2, where half the patients were on placebo and half the patients were on Rusfertide. And what you see on the left is the full analysis population. So this involves all the patients who were actually randomized. Some patients during the clinical hold did not come back, so they did not complete part 2. So if you look at the result on the right is all the patients who were randomized, even though they didn't complete part 2. That's called an intent to treat analysis. In both of the analysis, the response rate is very much higher in Rusfertide, 60 to 69% compared to about 17%. So what does response mean? Response means having the hematocrit controlled below 45 and not meeting eligibility criteria for a phlebotomy. So very, very effective drug in controlling the hematocrit so that the patient no longer requires or meets criteria for phlebotomy. And I think in the Rusfertide arm, only 3 patients actually got phlebotomy. So how long does this treatment work? This is a follow-up from the American Society of Hematology. And what you see in the orange is the hematocrit control in all the patients who are on Rusfertide. We have data at 2.5 years in this study, and you see that the hematocrit control effect goes for years. The part that goes up right here, this is placebo. So the patients in placebo, the hematocrit goes up. But then when they come back on Rusfertide in part 3, the hematocrits come back under control. I need to mention very quickly that this part that we published here, all this data was published in the New England Journal of Medicine with Dr. Marina Kremenskaya, who's a participant here today as the lead author of the publication, and she also presented this data at the European Hematology Association last June. So just want to acknowledge her contributions and the contributions of the Mount Sinai and other investigators in the United States. So we were able to get some data on symptoms. And in the part 1, which is this part here, just to remind you. So this is the data from part 1, where all the phlebotomy requirements go down and the hematocrit's controlled. In patients who had moderate to severe symptoms, we see improvement from baseline in the levels of fatigue, in the feeling of early satiety, in inactivity, problems with concentration, night sweats, and itching, the pruritus. And then the question is, well, what happened in part 2 when half the patients went to placebo and the other half in Rusfertide? What happened there is that half of the patients in placebo withdrew because they either had symptoms or required phlebotomies, and the protocol allowed the patient to withdraw at any time from the randomization. They all wanted to go to part 3. So all the patients who withdrew, even some patients who were on drug withdrew to go, like, remember our response rate, like 60%, 70%? Some of those who did not respond because they withdrew, and they wanted to go to part 3 to just make sure they were on drug. The phase 3 is very different. The phase 3, if you withdraw early, you cannot go to the open label extension. Because what happened because of the withdrawal is we don't have PRO, patient-reported outcome data, from part 2. And that's really important when you compare the placebo to the Rusfertide. Here, we're just comparing patients from baseline to the time of the analysis, but a reviewer can argue, well, they all know they're on Rusfertide, so maybe there's a placebo effect. How do you rule that out? So we need the data from the randomized study. But this also has been very encouraging. We've heard anecdotes from patients who could not work and were able to return back to work. And we'll put some of these patient vignettes together, but investigators have shared wonderful stories about patients who derive benefit. So here, let's get back to the ferritin. Ferritin basically is iron that's in a protein that's being carried through the body. And at the start of the study, all of the patients have very low ferritins. That's consistent with iron deficiency. And then while on treatment, the ferritin goes up into the normal range, above 100, but it doesn't go very, very high. It goes to the normal range. And we think that this might be one of the reasons patients are feeling better, is that even though Rusfertide is blocking iron delivery to the bone marrow, it is allowing some iron delivery to other parts of the body. And I don't have a slide for it, because I could go on and on with a lot of slides, but in the Pacific study, in patients who are iron deficient in their iron levels in the blood, when you treat them, in those patients, we see that the iron goes to the normal range. So we are getting evidence that we are reversing the iron deficiency, not just with the ferritin, but actually the serum irons. And in the patients who are iron deficiency, there's something called the MCV, the mean cell volume, the volume of the red blood cells. A lot of patients have red blood cells that are much smaller. They're not as healthy as patients who don't have PV. In those patients, the MCV becomes normal. So we're analyzing that data in the REVIVE study. The Pacific study is only 20 patients. Hopefully we'll have some of this data for you at ASH, but I think another effect of Rusfertide may be that it's making the red blood cells a little healthier. Not just controlling the hematocrit, but the red cells that are being produced might be a little healthier because now the MCV is becoming normal. So what are the side effects? The key side effect that most patients get is an injection site reaction. So it can be redness, it can be pain, it could be itching, it could be a little induration or swelling. It's very common, but what we hear, and we plotted it out, is that over time, this decreases in frequency and in severity. And then a lot of the other symptoms, fatigue, pruritus, headache, dizziness, those tend to be effects that you see with polycythemia vera. Some patients get a little bit of anemia, but we correct that by giving the right dose. Some patients, you go up on the dose. If they get a little anemic, you cut the dose down, the hematocrit comes up again. So as we continue to follow the patients, we do not see any new safety signals. The serious adverse events have occurred in 20%. A lot of these are skin cancers that I've mentioned. Patients with PV have a high rate of skin cancers, and that rate can go up when they use medications like Hydrea. In this study, before the patients ever got treated with Rusfertide, 27% of them already had another cancer. So it's a reminder for PV patients to get periodic skin exams because this can happen without Hydrea, but the risk goes up on Hydrea or on ruxolitinib. And then we have seen a total of 5 thromboembolic events, and these occurred in patients with risk factors or patients who had prior thromboembolic events. In patients who are low-risk PV, we have not seen any thromboembolic events with longer follow-up. So this is still early to make conclusions about whether we improve the thromboembolic event rate, but we haven't seen increased risk of thromboembolic events. And in our other studies that we have done in beta thalassemia and hemochromatosis, we haven't seen any thromboembolic events. And we also haven't seen any skin cancers in those other studies that we have done. The skin cancer risk seems to be primarily the patients with PV. And so what's happening with REVIVE? Most of the patients in the REVIVE study are finishing the study. So by June, all the patients on REVIVE will have completed the study. But what the patients are telling us is, can we keep them on Rusfertide? They want to stay on Rusfertide. So we are opening up a new study called THRIVE. And right now, this study is only for the patients who are graduating from REVIVE. And we have about 47 of the patients who have been on treatment now for over 2+ years who are signing up for another 2 more years. And we interpret that to mean that the patients feel good while they're on the treatment, they feel better, and they want to stay on treatment. So I know I've gone on a little bit long, but this is just a schema of the VERIFY study. We just finished enrolling, and some of our investigators from the study are here, and want to thank all the investigators. Some of the patients from the study are also here. Want to thank them as well for their participation. So this is very different design. Same endpoints though. The endpoints are very similar, and we have a lot more patient-reported outcomes as secondary endpoints. But basically, from the get-go, they get randomized to placebo or Rusfertide. They have to stay on for 32 weeks, and here we will do an efficacy analysis, and we'll do analysis of the patient-reported outcomes. The FDA wants durability of response, so we monitor the patients for 52 more weeks, and then we offer 2 years more of treatment. So important message for patients is that all the patients on placebo will go to Rusfertide here. So everybody gets Rusfertide. They just have to finish this part. If they drop out of this part, they cannot come over here, and they cannot come over here. So this study just completed enrollment, and we're hoping to announce results in the first quarter of 2025, because we need 1 year of treatment data to submit to the regulatory agencies. So with that, I think I've already mentioned the summary, and just want to thank all the principal investigators and the patients who have participated, and happy to answer any questions. Thank you.
First of all, I want to just clarify one point. As a patient organization, we should be neutral. And then sometimes people ask me, why are you letting talk, an owner of a company who has a specific product, which is not even registered in Spain, which is correct. But, we will offer any information from any pharmaceutical company who would like to do that through our channels. So I think the information which we can obtain directly from the industry, directly from the people who have been working in these products, is very, very valuable for us. We have to know it. I want you to know that. And this is why I let these people talk. And it's, sometimes people tell me you're married with them because of financial interest. Obviously, in order to organize an event like this, you need a little bit of money. And if you make it in Barcelona, you need a lot of money. So obviously, we receive money from the pharmaceutical industry, but we have our own opinion. We have our own way of thinking. Whoever from the industry wants to come here to talk, they can talk and they can give his opinions, and we will answer. So I just wanted to make this very clear, that we are not giving any preference to anybody. Here in this case, we have Christina, who is she's experienced with product. She's in a clinical trial, And having now the possibility of a person here who makes it, maybe you can explain us what is the experience. You will have to put your earphones on, yeah? She will talk in Spanish.
Sí, bueno, yo les cuento un poquito mi caso. Yo empecé hace 10 años diagnosticada de trombocitemia esencial y hace 6 meses, bueno, pues me comentaron los hematólogos que no era trombocitemia esencial, que era policitemia vera. Me hicieron una biopsia de médula, me habían hecho una anteriormente al principio de la enfermedad, pero por lo visto no fue concluyente. Y en 10 años no tuvieron ganas de volver a repetirla, debe ser, porque no me volvieron a hacer. Entonces, cuando ya me dan el diagnóstico de PV, yo bueno, pues hablo con Peter y Peter me recomienda lo primero que me vaya a un especialista de enfermedades MPN para, bueno, pues para realmente estar controlada, y también me ayuda a conocer Rusfertide y a poder participar en el estudio. Mi experiencia, la verdad, que es muy buena, porque yo me había hecho 3 flebotomías seguidas y no había conseguido bajar el hematocrito. Y además las flebotomías en mi caso eran muy desagradables porque debo de tener la vena muy fina, la sangre muy espesa, pero no sale bien la sangre, entonces pues eran dolorosas y eran costosas para mí. Y a través del el tiempo que llevo con Rusfertide, que son 3 meses aproximadamente, pues quitando los picores que produce la inyección, que bueno, son tolerables, sí he notado mejoría y sí he conseguido que el hematocrito se mantenga, bueno, ahora mismo está en 41. Y bueno, pues esa es mi experiencia. La verdad que muchas gracias a Rusfertide. Y bueno, y animo a la gente a participar en los estudios clínicos, porque no solamente por ellos, sino por todo lo que viene detrás, ¿no? Cada vez vemos más gente joven, más niños, y creo que bueno, que si entre nosotros nos echamos una mano, esto no avanzaría. Y bueno, nada más.
Does anybody have a question on the Rusfertide? I mean, anybody have would like to have more information?
I'm not sure how to ask this question. I'm not sure if it's a good question. And if it's a good question, I'm not sure I get the good answer. To me, it seems like if you explain the working of PTG 300, it should be beneficial to all PV patients, not only the ones requiring phlebotomy. Nevertheless, there is a lot of talk about the application of your drug when there is a lot of phlebotomy. Is this because this is the most targeted, best group requiring this medication? Or is it because you're looking for a niche that distinguishes yourself from the other drugs that work on PV?
It's a good question.
I think
For the good answer.
no, generally there's not a lot of phase 3 trials that have been done in PV, and it's not uncommon to follow a strategy where the FDA or the European health authorities is going to recognize the unmet need. So you tend to start with a patient population where the unmet need is more striking. I've worked in prostate cancer before, and you start out with the very end stage and you work up to much earlier stages. Here, this is patients who are receiving standard of care. The doctors and the patient have decided what treatment to be on. Despite the standard of care, they're still requiring a lot of phlebotomies. And we're not telling the physicians you should be managing this way. physicians have decided with their patients that this is how they're being managed, but the phlebotomy requirement is very high. So this is a way to a regulatory approval, but in the future, I would anticipate that either the company or investigators or cooperative groups in some type of trial would want to address other populations, patients who don't require so many phlebotomies, only low-risk patients. Like in low-risk patients, that might be, I mean, the low-risk patients would be another patient population to focus on, just like other sponsors have focused on that population. So I think this is generally how drug development works. If it works in the patients who have the most unmet need, then you can move on to areas where or patient populations where it might take us a little longer to get an answer. I hope that was a good response to your question.
I wanted to ask you, I mean, a patient who goes into a trial with you has to fill in a lot of paper, which is part of the deal. And I know that you're putting very interesting questions. Do you have priorities? Do you have this and that? I mean, if you're asking if you have priorities, do you want to see if your product eventually, if it takes it for a longer period of time, it could help the whole priorities effect?
Yes, we listened to the agency, FDA, and European health authorities, and we are using the currently available patient-reported outcome tools, but there is no specific form for just PV. There's the MPN tool, and then there's a myelofibrosis tool. Unfortunately, we were asked to focus on the MF tool, but the symptoms of MF and PV are pretty different. And so part of what we've done is we've interviewed patients with PV who are not on the study, but would meet criteria, and they've confirmed what we're seeing here on what is important for them. So we'll be going to the FDA and discussing, prioritizing the symptoms that the PV patients are telling us when we analyze these tools. Because there's some symptoms from the MF patient-reported outcome surveys that don't really apply from MF to PV. But that is one of the reasons when patients participate in this trial, they have to fill out a lot of forms. Because sometimes the FDA wants a 24-hour recollection at different time points over the past 24 hours or the past 24 hours. Sometimes they want you to respond over the last week. We're also doing quality of life in addition to patient-reported outcomes. But the symptoms that patients have told us consistently are fatigue, problems with concentration, and itching. Yeah, for polycythemia vera. And we're going to hope that the FDA allows us to prioritize those patient-reported outcomes in the way you rank them in order of priority in the assessment of the tool.
Great.
Okay.
Thank you very much.
Thank you.
Thank you.